Okay, hi everyone. This is our lecture on chapter 7, which is the chapter for on photosynthesis. So I always joke that it's, everyone's favorite chapter. Um, but anyway, no, I'm nonetheless a super important process to learn about. Um, a lot of the concepts that we're going to learn about in this chapter will then apply to the next chapter for cellular respiration. So I feel like it's always a little bit rough getting through this chapter, but when we talk about cellular respiration, things just become a little bit more clear. Okay, so in terms of the outline, broken down, just to talk a little bit, just sort of in general about photosynthetic organisms. This is probably something that I've already talked about in other chapters. Then we'll talk about the process of photosynthesis, talking about converting solar energy, right? Specifically that part of it, which is really the light reactions. And then this part, the fixing the carbon dioxide, that's really the Calvin cycle, or the dark reactions. Sometimes you'll hear that being called. There is a last section on other types of photosynthesis. I don't hold you guys responsible for that section. I just leave it in here just in case somebody's interested in learning a little bit more about it, but you're not responsible for that last section. Okay, so I'm not even going to really go over that section. All right. So just to start off with a little introduction, again, this is the introductory sort of couple paragraphs that the book always has, and it's the sort of why should anyone care about photosynthesis, right? That's essentially what these introductory paragraphs are doing. And so here, this is talking about photosynthesis as being a key to solving our fuel crisis. Okay, so there's just two examples there of basically scientists being able to use genetic engineering that not, I don't want to say interferes, but that affects the genetic engineering, or the genetic changes that are induced are actually going to affect photosynthesis, right? And so for the example here with this camelina, okay, this is a drought resistant oilseed crop. And basically, again, via genetic engineering, you can improve the efficiency of photosynthesis. Okay. And so one of the ways I think they do this is sort of modifying the leaves that are on the top of this plant to make them lighter, so that then those lower leaves can actually take in the sunlight, and then that will make that overall process much more efficient. Also, they're working on improving carbon dioxide absorption, right? So if the plant is able to take in more carbon dioxide, it will then be able to make more carbohydrates. And again, these can be used as raw materials for biofuels. Okay. Another example is terpene. And so this is an organic molecule that's made from pine trees, right? So when they photosynthesize, this is a molecule that can be made, and then that's used to make turpentine. And basically, by increasing this terpene production, you can then use this again to make diesel and aviation biofuels. Okay, so just tweaking this process of photosynthesis can actually really increase our production of biofuels. Okay, so one of the reasons why you should care about photosynthesis. All right, so let's talk a little bit in general. Again, I think this is some stuff that we've probably mentioned before. So we've definitely talked about the idea of sort of cycling of energy, right? And that really all life on Earth depends on solar energy, right? That photosynthetic organisms can take in that solar energy and convert it into usable energy, and then use that energy to make carbohydrates. Okay, and remember that when we talk about photosynthetic organisms, we're not just talking about plants, right? That's usually what we think of, but there are algae that photosynthesize, and also there are photosynthetic bacteria called cyanobacteria. Okay, we call these types of organisms autotrophs because they're able to produce their own food, right? Auto, own, or self. Okay, so they can produce their own food. If you talk about in general that process of photosynthesis, again, I think I've already said this probably a couple times, captures solar energy, transforms that solar energy into chemical energy or usable energy, and then that energy is used to make carbohydrates. Those carbohydrates then provide a source of stored energy, right, for heterotrophs that then eat them, right? So heterotrophs like us and other animals eat those carbohydrates. Our body then is able to break down those carbohydrates to generate ATP for us to use. Okay, so a couple terms here, right? Autotrophs, heterotrophs. Again, heterotrophs are consumers, another word for them, because they are not able to make their own food. They're going to ingest food like carbohydrates that are made by plants. Okay. All right, I already said this too. It says both autotrophs and heterotrophs use organic molecules produced by photosynthesis as a source of chemical energy for cellular work. So I gave that example. Heterotrophs eat the carbohydrates that are produced by autotrophs, right? And then break that down. But autotrophs themselves need to utilize some of that energy to basically perform other types of chemical reactions in the cell. Okay? All right. So this is, again, we've pretty much seen a similar figure like this before in just various different ways. And so this is really showing you the sort of cycling of energy in an ecosystem, also the relationship between autotrophs and heterotrophs. Okay, so this all should be pretty familiar. It's basically what I just explained on that last slide. Here's just showing a picture of Oscillatoria, right, which is a cyanobacteria, right, a photosynthetic bacteria, kelp, and algae, and then obviously these large sequoias, which are huge trees, right? Okay. All right, so if we talk about where photosynthesis takes place, right? It's in the green portions of plants, okay? So particularly in the leaves of plants for the most part, and within the leaves, there's tissue called mesophyll tissue. So it's within those cells and that mesophyll tissue, those cells have chloroplasts, and that's where photosynthesis takes place, okay? The raw materials that are necessary for photosynthesis, again, we've talked about this before, are carbon dioxide and water, right? Sunlight as well, but the plant is going to need to take in carbon dioxide and water as well. Go ahead, buddy. Sorry, I'm letting my dog. Go ahead, bud. All right, never mind. Okay, so in terms of how the plant accesses those raw materials, right? Water is going to come in through the roots, will move up through that vascular tissue and get to the leaves, right? Carbon dioxide will actually enter through the leaf in small openings called stomata, okay? And then it will diffuse into the chloroplast. Carbon dioxide is not going to come in from the roots because carbon dioxide is in the atmosphere. The thylakoid membranes in the chloroplast, right, contain chlorophyll. Okay, and also other pigments. We'll talk a little bit about chlorophyll, or a little bit more about chlorophyll as a pigment and other pigments as well. But these pigments are able to absorb the solar energy. Okay, so the pigments are necessary to absorb that solar energy. That solar energy then gets converted into that usable energy. But without the pigments there, like chlorophyll, the plant would not be able to take in or absorb that solar energy. Okay? Once that solar energy is absorbed by those pigments, basically it energizes the electrons in those pigments. And then it basically, those energized electrons get ejected and they end up going to an electron acceptor. Okay. And then they get passed down through an electron transport chain. And then that's how energy is produced. Okay. So we're going to talk more about all of these things. All right, the carbon dioxide again is taken in, and that's actually reduced to form a carbohydrate. Okay, so this kind of up until this part, we're talking about the electrons. This is really all of this process that we're going to talk about in more detail is really to generate energy here, right? The carbon dioxide that's coming in through the leaves is what's going to get fixed to form the carbohydrate. Okay, and so that's what this is saying here. All right, so just a little bit, a picture to show this. So again, we're talking about where photosynthesis occurs in the plant. And for the most part, when you think about where photosynthesis occurs, it's in the leaves. Leaves have lots of surface area, right? It's within that mesophyll tissue where you have these cells that have chloroplasts, right? And if you zoom in on the chloroplasts, right, photosynthesis is going to happen at the chloroplast. This is a typo, right? This is stomata that we talked about on the last slide, where the carbon dioxide is going in and out, or carbon dioxide going in, oxygen coming out, these openings in the leaves, okay? Don't confuse that with stroma, which is the fluid like substance kind of inside the chloroplast. Okay, so again, we should know the components of the chloroplast now. I told you, in chapter 4, you didn't have to worry about it because now we're going to talk about it and talk about what part of photosynthesis takes place where. Okay, so we should know the parts of the chloroplast. remember it has a double membrane, okay? Here, if you want to talk about this sort of in general, they're grana, multiple granum is singular, these stacks of thylakoids, but we'll zoom in on the thylakoids, right? The thylakoid membrane and then the thylakoid space. So the light reactions, which we're going to talk about first, take place in the thylakoid membrane. Okay, so anyway, this is sort of becomes important now because we're going to talk about where these different parts of photosynthesis take place in the chloroplast. Okay. All right, so like I said, the light reactions, right? Basically two main parts of photosynthesis. The light reactions are called the light reactions. They take place in the presence of light. Okay, those are the energy capturing reactions like I mentioned, right? Chlorophyll absorbs the solar energy. Electrons move down an electron transport chain. And within this process, you're going to generate ATP. Okay, we're going to talk about these details again, all right? The Calvin cycle reactions, which take place in the stroma, okay, by the way, these light reactions again take place in the thylakoid membrane, okay? Calvin cycle take place in the stroma. This is where the carbohydrate is made, okay? So carbon dioxide is reduced basically to form a carbohydrate. So the energy that's produced in the light reactions is used to drive the Calvin cycle reactions, right? And to produce the carbohydrate. Okay, it's named, and the reason why it's called Calvin cycle, it's named after Melvin Calvin. Okay, so if you remember in the last chapter, in chapter 6, we talked about oxidation and reduction reactions. Okay, a lot of the reactions that occur in photosynthesis involve oxidation and reduction. Okay, remember oxidation is the loss of electrons or the loss of hydrogens, right? Hydrogen ions, and reduction is the gain of electrons or the gain of hydrogens. Okay, if you talk about in general, and I actually showed you this, right? The general reaction for photosynthesis in that last chapter, carbon dioxide is reduced, right? And water is oxidized. Okay, so if you want to look at this picture here, what this is showing you, this is an aquatic plant, Elodea. Actually, that's one of the pictures that we showed in terms of when we were talking about diffusion, right? Or particularly osmosis. It was basically a microscopic picture from the leaf of an Elodea. So this is an aquatic plant. What you see here is these bubbles that are being released are oxygen, right? So it's oxygen being released. And so you can visualize this. You can do this actually in the lab. If you put Elodea in some carbonated water, you shine a light on it, you'll see these bubbles being emitting from the plant. Okay, so I like this slide, right? This overview slide is super important. I kind of just went through this in words, but let's go through it again. If you do not understand the overview and what the function is of the light reactions and the dark reactions, or the light reactions in the Calvin cycle and how it fits together, all of the other details don't really matter, okay? So super important to understand this slide, right? All right, so again, you have solar energy coming in. Where is it particularly coming in? Into the chloroplast, but really into these thylakoids, right? Into that thylakoid membrane. Okay, and so again, it's the light reactions, it's chlorophyll, these pigments that are part of the light reactions that take in this solar energy. You can see water's going in here. And basically, what is the output here? It's energy, right? It's energy in the form of NADPH, and it's energy in the form of ATP. Okay, here also oxygen is released, but that's essentially just a byproduct. Okay, so solar energy came in, water came in, and energy was produced. Where's that energy going to go? It's going to go into the Calvin cycle to drive the Calvin cycle reactions. Remember here, carbohydrates are being made, right? This carbon dioxide coming in is going to get fixed into a carbohydrate molecule. Remember, when you think about synthesis of any type of molecule, right? Synthesis in general, usually those reactions require energy, okay? So we're synthesizing carbohydrates here. It's going to require energy. Okay, where does that energy come from? It comes from the light reactions. So NADPH, ATP go into these Calvin cycle reactions. So in order to drive those reactions to make carbohydrates, okay? This is, and we'll talk a little bit more about this, but essentially this can then be converted into glucose, okay? So this is carbohydrate, okay? Where is it taking place? In the stroma. Where are the light reactions taking place? In the thylakoid membrane, all right? So again, super important to understand this. If you understand this, you're good. You just sort of plug in some of the details that we're going to talk about. But this is really probably the most important slide of the whole presentation. Okay, so I said we were going to talk a little bit more about those pigments, right? So this is particularly, again, focusing in on converting the solar energy, right? The pigment's job is to basically absorb that solar energy, right? And then convert it into usable energy, okay? So it also says photosystems here, so we're going to talk a little bit about the photosystems as well. And so these are essentially, complexes, okay, including the pigments. And so we'll come back to that in a second. Let's just talk about pigments in general. So these are chemicals that absorb certain wavelengths of light, right? Okay, so they absorb certain wavelengths of light, and they also reflect certain wavelengths of light, okay? So a pigment will absorb some wavelengths of light, but then it will reflect others. And so basically what you can do is you can make an absorption spectrum or look at an absorption spectrum of a particular pigment, right? So you want to see, it'll basically show you what wavelengths of light are absorbed by that particular type of pigment, okay? All right, so that's what it says. Pigments found in chlorophyll absorb various portions of visible light, all right? So like I said, this absorption spectrum is going to show you that relative absorption, okay? Now, chlorophyll, as we know, is green, right? Why does it look green? Because it absorbs much of the reds and the blues, right? Those wavelengths, and it reflects the green wavelengths, okay? So that's why it looks green. Other types of pigments that exist, these carotenoids are other accessory pigments. These will absorb light in the violet, blue, green range and reflect yellow and orange light. They look yellow-orange, okay? All right, so if you look at these graphs here, these are these absorption, spectrums that we're just talking about, right? So one, this is just showing you this visible light, right? So if you look at light, energy in general here, right? You can see, X-rays, UV, infrared, visible light is within this orange spot here. And if you expand on that, you can see these are the different wavelengths or the range of wavelengths of visible light, okay? And so now if we then sort of put the pigments that we were just talking about, sort of along these wavelengths, right? And we're going to talk about relative absorption, right? So which wavelengths are absorbed by certain pigments, okay? So if you look at chlorophyll A and B, you can see that these blue and purples, right? They have high absorption. When you get to the greenish range, very low absorption. These are green, right? They reflect these wavelengths of light. They do not absorb it, okay? And then you can see then they're going to absorb in the orangey and red range. If you look at the carotenoids, they're going to be absorbing much of this wavelengths in the blue and the greens, but you can see they have no absorption down here in this end because they are orange, okay? This is not something that, nobody, you need to memorize or anything like that, but, be able to recognize this. I mean, I guess if I showed you this, you should be able to interpret what it's showing you, okay? So remember, chlorophyll looks green because it reflects green light, right? It reflects these wavelengths, around between 500 or around 500 wavelengths, okay? Not because it absorbs those, but because it reflects those. All right. Now, we have to talk in more detail about these light reactions, okay? So, like I said, that overview slide, super important. Now, we're going to talk about some details. And actually, I think that overview slide comes back. Let me just make sure. Yeah, okay, good. I think I put it back in there. So anyway, I'll come back to that to kind of show you where we're at. But again, light reactions are called light reactions because they happen during the sunlight, and they are the reactions that take in that solar energy, that sunlight, and convert it into usable energy. Okay, that's what's happening during the light reactions. Remember that overview slide that I showed you. Now, the non-cyclic pathway is what's going to happen most often. Okay, that's what we're going to talk about. We're not going to worry about going through the cyclic pathway, but I have to tell you that it does exist. So the non-cyclic pathway, which is what we're going to focus on in terms of detail, does not happen every single time, because sometimes basically the cell has to reroute an electron, okay? Because ATP and NADPH are not made in equal amounts. Again, if that just like sounds like gibberish to you, then just don't even worry about it, all right? But again, I have to just tell you that that does exist, but we're going to focus on that non-cyclic pathway. Okay, so we know that the light reactions capture the light energy, and they do this with two photosystems, okay? So there are two photosystems, photosystem 1 and photosystem 2, okay? A photosystem is just a pigment complex. Okay, so we talked about the pigments before, and that pigment complex basically collects the solar energy. So it acts like an antenna to collect that solar energy. And then there's going to be a particular part of that pigment complex called the reaction center. That's the part that's going to become excited, and those electrons are going to become excited. And then ultimately those electrons are going to be sent to an electron acceptor, okay? The photosystems are located in the thylakoid membranes. Again, the light reactions occur in the thylakoid membranes. All right. Again, we're focusing on the non-cyclic pathways. They produce ATP, okay? Both cyclic and non-cyclic. Okay, but so for the light reactions, ATP is produced and NADPH is produced. Okay, both of these are energy-rich molecules that are then going to be used to drive the Calvin cycle. Okay, so the light reactions produce energy. That's what they do. All right, now there's a whole bunch of words here. Okay, I'm not going to read through every single one of these words. I'm going to flip to the diagram and basically explain it with that picture there, okay? But what's on this slide in words, okay, explains what's happening in this diagram here, okay? So again, this was our overview, right? We're talking about the light reactions here. Solar energy coming in, we have everything taking place in the thylakoid membrane, right? And we're producing energy, okay? And this is supposed to be O2, not just an O there. All right, so let's specifically look at those light reactions. So now we're zooming in on those light reactions. So remember we said these photosystems are just pigment complexes, okay? So here's your photosystem 2. The reason why it's 2, even though it sort of comes first in the sequence, is because it was discovered second, okay? So your solar energy, right, is coming in here. And basically, again, this pigment complex acts as an antenna for this solar energy. Ultimately, one part of this is going to become the reaction center and become excited, right? It's going to then give off its electron to an electron acceptor here, okay? One other thing to note is that these pigment molecules here are giving off electrons. Well, they need to then be replaced, okay? So we said water is required, right? Coming in here to the light reactions. Why? It's actually the electron source here. So once this electron goes to the electron acceptor, we have to replace that lost electron. How is it done? It's by the splitting of water. Okay, so basically just know that this electron is coming in to replace the one that gets sent to the electron acceptor. Okay, all right, so we move on from there. Once this electron acceptor gets the electron, it then passes the electron down through a series of carriers, and we're going to talk about this electron transport chain in more detail in a second, so don't worry about the detail right now. But basically the electrons it's called the electron transport chain, they get passed along from carrier to carrier in a stepwise fashion, and in the process you have hydrogen ions that are released and that they form this gradient that then ultimately leads to the production of ATP. Okay, so the ATP is produced from the electron transport chain, but in an indirect way. We will focus on this electron transport chain and explain how exactly that ATP is being produced. Okay, so right now we're not showing those details. All right, where's that ATP going? To the Calvin cycle reactions, right? To drive those reactions. Now we also have to talk about photosystem 1 over here, right? And the production of NADPH. So photosystem 1, just like photosystem 2, is a pigment complex, okay? Solar energy comes in, it's going to, these are going to absorb that solar energy. Particular pigment molecules are going to become excited. That's the reaction center, going to give off an electron, right? And then goes to the electron acceptor. This then gets transferred directly to NADP, right? And if it's gaining an electron or gaining a hydrogen here, right? Remember we talked about oxidation and reduction, right? Oil rig, oxidation is loss, reduction is gain. What's happening here? This NADP is gaining a hydrogen, right? Okay, so anyway, this and for photosystem 1, basically there's no electron transport chain. It's just this conversion of NADP plus to NADPH. That also goes into the Calvin cycle reactions and acts as an energy source to drive those Calvin cycle reactions. Okay, so that is the light reactions. Now we have to talk more about that electron transport chain. Okay, so that's this here. We're continuing on, kind of, again, lots of words on these slides. They're there for you so that when you go back and study, you can read through those words. But everything that these words are describing is also in the figures, right? Okay, so I said the electron transport chain. If we go back here, here's the electron transport chain. Okay, so we have to talk more specifically about that. And basically, it consists of protein complexes, right? So cytochrome, protein cytochrome complexes, and another type of protein here, plastoquinone. You guys don't need to know specific names of these proteins, okay? But basically, these are carriers, and so basically, these are carriers that will move these electrons from complex to complex, right? So this is the plastoquinone and the cytochrome. They're basically acting as these carriers, okay? So the electrons basically move, right, through these carriers, okay? That's essentially what's happening. So you can see these electrons here. This is electron transport chain. Again, they're being just transferred from molecule to molecule, okay? And as they get transferred, what happens is hydrogen ions are released, okay? These hydrogen ions then accumulate in this thylakoid space, okay? So see that here. Hydrogen ions accumulate in the thylakoid space. Okay, so that's what this is saying here. Right? Okay, and now we're going to talk about this ATP synthase. Okay, so again, for our purposes, let's just think of this as electrons are basically passed from molecule to molecule, okay, through the electron transport chain. Each time they're passed, basically hydrogen ions are released. These hydrogen ions accumulate in the thylakoid space. Now, what does this do? This sets up a steep concentration gradient, right? Remember we talked about this. We talked about diffusion of molecules. So if you have a lot of hydrogen ions inside here and not a lot outside, these hydrogen ions are going to move along their concentration gradient, right? They're going to have the tendency to go this way, out. The only way these hydrogen ions can get out of that thylakoid space is through something called ATP synthase. This ATP synthase is just like any other membrane channel, okay? This is the thylakoid membrane, okay? But just like we talked about the plasma membrane and different types of carrier proteins and channel proteins that exist, same idea here. This just has a special function. So one, its function is to basically transport those hydrogen ions through the membrane, right? But once those hydrogen ions move through the ATP synthase, each time a hydrogen ion goes through, ATP is produced. Okay, so that's why I said that ATP is sort of produced indirectly from this electron transport chain, because what's actually happening is the hydrogen ions are released, right? They then accumulate. Because they accumulate here, and there's not a lot out here, it sets up a steep concentration gradient. These hydrogen ions have the tendency to move this way, right? The only way they can get through the membrane is through this ATP synthase. When they move through ATP synthase, ATP is then generated. Okay, so that's how the ATP is actually generated. And again, like I said in the beginning of the chapter, this same process is going to occur in cellular respiration. Okay, so when we talk about the electron transport chain, when we talk about cellular respiration, which is the next chapter, and we're talking about in the mitochondria, you're going to see lots and lots of similarities. Same process, okay? And we call this chemiosmosis, okay? These hydrogen ions moving and then producing the ATP, okay? So again, that may take a little bit of kind of time to sort of have that sort of sink in. But again, what I really take home message is that we understand that you know, the ATP is generated from the electron transport chain, but that it's indirect. How it actually happens is this here. Now, a lot of times on the exam, I will put a diagram like this on there and ask you about it. Again, I'm not going to ask you to just like blatantly or sort of without any sort of prompt tell me exactly what's going on, but we should understand that you know, this term chemiosmosis is basically the production of ATP due to these hydrogen ions moving through the ATP synthase. Okay. All right. So that's what this is telling you here. All right. Okay. So I think we already, yeah, this is all kind of the same. All these words here are basically describing what I just described to you here. Okay, so remember, there's a lot of words surrounding these diagrams. They're just basically going through what I just went through with you with that figure out. Okay, so you can take a look at that. And again, I purposely leave so many words on the slide so that you guys can study from these slides. Okay, so again, it says this method of producing ATP is called chemiosmosis because ATP production is tied to the establishment of a hydrogen ion gradient, meaning this, right? High concentration of hydrogen ions here, low concentration here, okay? They have the tendency to go this way. They move through ATP synthase and generate ATP, okay? All right, so now that brings us to the next part of photosynthesis. Remember, we can divide photosynthesis into two main parts: the light reactions and the Calvin cycle. Okay, we just went through the light reactions. The purpose of the light reactions is to produce energy for the Calvin cycle. Okay, the purpose or the function of the Calvin cycle is to make carbohydrates. Okay, so the Calvin cycle is called a cycle because it is a cycle, right? It's a cyclical series of reactions. It's going to take in carbon dioxide from the atmosphere and produce carbohydrates with that carbon. Okay, we call this C3 photosynthesis because there are other types of photosynthesis. Remember, I mentioned that at the beginning of the chapter. And I told you you're not really responsible for it. Okay, so if you see it called C3 photosynthesis, that's because there also is C4 photosynthesis. There's other types of photosynthesis, okay? But we're not going to worry about that. The three main stages of the Calvin cycle are, you should know these three stages: carbon dioxide fixation, carbon dioxide reduction, and Ruby P regeneration. Okay, those are the three stages of the Calvin cycle. All right, so let's talk about this Calvin cycle. Okay, and I'm going to obviously show you a nice figure of it. So we're basically hitting the high points of the Calvin cycle here. We're definitely not showing you all of the different reactions that occur. All right. So first it says, so carbon dioxide is going to come in, right? So it says plants as carbon dioxide fixers, carbon fixation, meaning carbon from the atmosphere, from carbon dioxide, gets fixed into a carbohydrate. Okay, that's where this fixation comes from. So carbon dioxide is attached to a 5-carbon Ruby P. Okay, and there's a specific enzyme that will attach the carbon dioxide, the carbon from the carbon dioxide to Ruby P. Okay. All right, and you end up with the 6-carbon molecule. That 6-carbon molecule then splits into 2 3-carbon molecules, and these 3-carbon molecules are called 3PG. This basically this is the reaction or the enzyme that catalyzes this reaction, right? So this is what this is telling you here. So again, now it's saying the carbon dioxide is now fixed because it's part of the carbohydrate. Okay, so that's the first part of it. So let's review that. Again, here's your Ruby P. So if you want to know the full name for these, you can look at them here. I don't expect you guys to memorize these, so I'm not going to be like, you know, Ruby P stands for what and expect you to write it out, right? I'm not going to do that. So it's here. So Ruby P, ribulose 1,5-bis phosphate, okay? 3PG, 3-phosphoglycerate, all right? So anyway, these are, I'm just going to refer to them by their abbreviated form, okay? Or in their abbreviated form. All right, so here we go. So you have, and so one thing to point out too, that you need 3 turns of the cycle to have a net gain of 1 G3P, and so that's why you're seeing 3s here. Okay, not something to worry about or fixate on. Let's just understand the main process here. Okay, but that's the reason why you're seeing 3s. Okay, so 3 turns of this to generate 1 G3P. You're going to see that's going to come out over here. Okay. So just wanted to point that out in case you were wondering why. Again, there's our overview picture, right? We already just went through the light reactions. The light reactions produce the energy necessary to drive the Calvin cycle. Now we're in the Calvin cycle. Here's your carbon dioxide coming in, okay? It's going to join with this ruby P. All right, okay? It forms this 6 carbon intermediate. Then that splits into 2 3 carbon intermediates, okay? All right, again, you can see here, this is a part of this that requires lots of ATP, lots of energy goes in. You're going to now form BPG, okay? And then this BPG can get converted into G3P. Again, requires energy. So the NADPH going in, converted back to NADP plus. These are basically, this is input of energy into here, okay? So again, reminder of that. Okay, so now you have this G3P, right? 1 G3P is going to go out of this cycle, and that G3P can produce organic molecules like glucose, but other molecules as well. And we'll talk a little bit about that in a second. So glucose doesn't come directly out of the Calvin cycle, but glucose can be produced from this G3P. Okay, so this G3P is a carbohydrate that then can be used to produce other carbohydrates or other organic molecules, not even just carbohydrates, right? So it's again, G3P can then be used to produce carbohydrates like glucose, but also other organic molecules, okay? All right. Now, the rest of these G3Ps that are produced here, one comes out, the rest of them have to continue in the cycle, okay? So that then ruby P can be regenerated and the cycle can continue, okay? So if you divide or look at these 3 different parts that we talked about before, here's your carbon dioxide fixation. It's basically the initial part, right, where the carbon dioxide is coming in, joining with the ruby P to form this 6 carbon intermediate, right? Then it becomes reduced, okay, carbon dioxide reduction. You have this, basically the formation of these, what starts it is the 3PG, right? 2 3 carbon molecules. And then basically as you move through the reduction, right, you're going to produce G3P. So basically kind of, you want to, I guess, kind of think about what's going in, or what the, really, I guess, let's think about what the function is of each one of these phases, right, that I just sort of went through, okay? And again, there are many other reactions in between some of these, okay? So we're highlighting some of the main parts of this process. All right, so in the reduction part, you're basically moving from these 6 molecules here, right? Of these 3 carbon molecules, C3, that's what this means, and then converting it into BPG, and then eventually into G3P. Then this G3P gets regenerated, right? Regeneration of ruby P gets basically, the ruby P is regenerated because this G3P gets converted into ruby P. Okay? Again, more ATP required. So basically all of the reactions that are occurring here within this Calvin cycle require an input of energy. All right. So, sorry, this is continuing on again. It's just like the other slide, right? I sort of went through the diagram in words. The words are on these slides that surround it to explain what's being shown in that diagram. Okay, so this is continuing on. So if you go, you go back from here, let's see, right? Carbon dioxide is now fixed because we talked about this 3PG. And then if you follow along here like we did, right, 3PG is reduced to BPG. BPG is then reduced to G3P, so that's why we call that carbon dioxide reduction, right? That's that part of it, okay? So again, we want to sort of know the main things that are occurring. They're named, those 3 phases are named based on what's occurring within those phases, okay? So this is carbon dioxide reduction because 3PG is reduced to BPG. BPG is then reduced to G3P, Okay. Okay. Remember, there's lots of, NADPH and ATP are required. All right. Again, so this is carbon dioxide reduction. Remember the first part. Again, this is describing the first part, carbon dioxide fixation. So it's that carbon dioxide coming in, joining with the Ruby P, right, to form this 3PG. Right? And then the last part, remember, is Ruby P regeneration. Okay? So this is just showing you here, zooming in on those basically some of these reactions here where you can see the input of ATP and NADPH. Okay, right here. Yeah, I mean, this pretty much shows you what's shown in the cycle. It's not really anything new. Okay. All right. Last phase, regeneration of Ruby P. What's happening, right? Basically, that G3P is going to be converted into Ruby P. Okay. And again, this is just talking about the numbers that you need. I don't focus on that. Okay, guys, I'm not going to ask you about how many turns it takes, but again, that's why you see those numbers there, right? Every 3 turns, you get 5 G3P that are used to remake the Ruby P molecules, okay? And so that's what this is talking to you about, this sort of 5 times 3, 3 times 5. Really don't worry about that, all right? I want you to know the main function of each of those 3 phases of the Calvin cycle that we went through. And what's basically happening at each one of those stages. And you can see it from this diagram, right? You can also read it in words from each one of these slides here. So again, carbon fixation, carbon dioxide fixation, carbon dioxide reduction, right? What's happening? And then the last phase, regeneration of Ruby P. Okay. And again, like I told you, I don't want you to worry about, the 3 turns and 3 times 5, whatever. I probably should just even get rid of this, but so you don't have to worry about that. All right. So again, this is just reiterating and focusing in on, this sort of reaction and that it requires ATP. Okay. And you can see that from that picture of the Calvin cycle. Okay, I started to mention this, right? So I said I would talk a little bit more about this. So this G3P that's coming out of that Calvin cycle, it can be converted into many other molecules, okay? It can form fatty acids and glycerol to make plant oils. It can form glucose phosphate to make simple sugars. It can form fructose. It can form starch and cellulose and also even amino acids, okay? So any biomolecules that are needed by that plant cell basically are derived from that G3P molecule, okay? So this is a little tree showing you that, right? Here's your G3P. It can produce any of these things, okay? It can produce glucose, fatty acids, amino acids, right? From the glucose then can produce more, complex carbohydrates, okay? There you go. All right, and as I said, you don't need to worry about this section. So I usually just skip it altogether. Again, I leave it in there in case you want to read about it, but there are other types of photosynthesis or sort of modifications to this process that we talked about. I mean, it's going to be the sort of same basic process, but there are, plants that live in certain environments that require this process to be modified, right, in order for them to survive. So there's adaptations that have occurred to allow these plants to survive in really dry environments and hot, dry environments and things like that. Okay, so that's what this is referring to. So you don't need to worry about that. All right, so that concludes chapter 7. Remember, most of the concepts that happen in photosynthesis are going to come back in the next chapter for cellular respiration. So the next chapter I find is a little bit easier to understand and a little bit easier to take because you've already gone through most of these things with photosynthesis. Remember, kind of, if you think about the overall reaction for photosynthesis and the overall reaction for cellular respiration, they're essentially opposites of one another. Okay, so that will be our next topic. Again, and I wish I remembered at the beginning of the lecture, but remember, you should have your exam 2 review sheet And you need to be filling in notes there, right? So maybe you're writing in notes in terms of, what's happening at each main stage of the Calvin cycle, right? Writing in notes for yourself to understand that so you can go back and use those notes to study for the exam. Exam 2 is going to be chapters 5 through 8, right? So chapter 5, 6, 7, and 8 are all included on exam 2. All right, thanks, guys.